Hello there. My name is Chris Hopkins. We got you here on 17 Minutes of Science. This is the show that explores the world of science and how it affects both the starting academic and the seasoned professional. Today I have as a special guest, Valeria Vásquez. She joins us from the Health Center, Sciences Center at the University of Tennessee. Hi, Valeria. How are you doing today?
Good, Chris. How are you? Thank you for having me.
Oh, well, thank you. On 17 Minutes of Science, we probe into a lot of questions, and I'd like to ask members of the audience if you feel free to please chat us a question at any time here. So as a quick intro for Valeria, she's an associate professor at the Health Sciences Center at the University of Tennessee. Her lab studies the mechanosensitive channels. Basically, dysfunction in these sensory receptors are implicated in a broad range of diseases, anywhere from heart arrhythmias to sciatica, chronic pain. And basically today's subject matter is going to be understanding the mechanosensitive channels through looking into C. elegans. And so with 17 minutes, and we're already got a minute, let's start in with some questions immediately for you. Valeria, can you tell us a little bit more about your research into mechanosensation and how you got started in all of this?
Yes, thank you again. So my lab studies how fatty acids modulate ion channel function. We are particularly interested in mechanosensitive ion channels, which are channels that sense force either through the membrane or the actin cytoskeleton. In my lab particularly, we study those that are modified by the membrane composition. I started this in my grad school. I did my thesis with Eduardo Peroso at the University of Virginia. Back then, I used to work with bacteria. So I used to work with the diet of C. elegans. And we were studying the prokaryotic mechanosensitive ion channels from a very biophysical point of view. So we were studying structure function and how these membrane pores sense the lateral tension of the membrane and they would open. So my interest started there. I learned electrophysiology, patch clamp, a little bit of microbiology. And when I was looking for my postdoc, I knew I wanted to continue studying mechanosensitive ion channels. And I happened to be in a meeting where Miriam Goodman was giving a sensational talk. And it really, really caught my eye because she was the only person working with, at the time, in vivo electrophysiology. She was doing EM. And, when I started digging into the C. elegans thing, I just fell in love of how amazing the techniques were back then and imagine now. So I asked Miriam to join her lab because at the time also we only knew about one eukaryotic mechanoreceptor complex that happened to be in C. elegans touch receptor neurons, and that Miriam and Martin Chaffee and Laura Bianchi and many others were piling on, a bunch of amazing data to demonstrate who were the subunits forming this mechanotransduction channel complex. So at the time, I wanted to combine what I knew with whatever was known in the field and try to understand if, at all, the membrane composition modified this mechanosensitive complex. So that's how I started working with C. elegans. And just, I don't know if you remember, Chris, but when I was about to go on my maternity leave, I was freaking out because I knew my project would be stalled for a few months. So Miriam heard about your company, I think Nudra back in the day, right?
Yeah, but Nudra probably back then. Yeah.
So we ordered a bunch of worms from your company because that was the way that we find, or Miriam found, to keep my project going while I was on maternity leave. So that was really nice.
We love to call that the outsourced postdoc service that helps people keep carrying forth and keep progress up.
That
awesome.
Really makes my maternity leave very relaxing because I knew the project was going. And then, so I finished my postdoc with Miriam and I moved to the University of Tennessee, where I'm currently at. Actually, my last week I received my tenure promotion.
Oh, so congratulations. That is awesome.
Since we started here, I have used, Nemametrics and in vivo because you guys are my postdoc away from the lab. And that has helped a lot. So we see elegans. So in the lab right now, I'm not working particularly to understand C. elegans, and I apologize to the worm lovers, but I'm more of I'm trying to use the beautiful PUFA or polyunsaturated fatty acid cascade of C. elegans, to use those genetic backgrounds or those genetic mutants to understand how polyunsaturated fatty acids affect mechanosensitive ion channels. So that's the in vivo part. Right now we're doing also mice behavior, which is way more complex than I thought, obviously. But we'll keep working with C. elegans very heavily.
Well, excellent. And that actually brought me to one of the questions I wanted to ask you is sort of what other approaches are you integrating with C. elegans? And so it's the mice would be a really interesting subject there. So, and a little bit just sort of what have you found about fatty acids? I mean, how impactful are they? How important are they for these mechanosensation channels?
They're super important because they embed the membrane with particular mechanical properties. So you can make a membrane really rigid if you have a bunch of saturated fatty acids, or less really rigid if you have polyunsaturated fatty acids. So we have done those measurements in Miriam and my lab with atomic force microscopy. And it's really amazing how that can impact mechanosensitive ion channel function. We also have determined that certain trip channels are very sensitive to the membrane mechanics. Not to say that they are mechanosensitive, but they are very vulnerable or sensitive to the membrane composition. And I would expect that every membrane protein would have certain degrees of sensing this membrane composition. So we use again C. elegans because the genetic background is really clean. So you can have either omega-3 PUFAs or omega-6 PUFAs or eliminate all of them. And then it's a really clean background that you don't have with mammals, right? So in mammals, we can modify the diet and check on that. But with C. elegans, genetically, we just remove the PUFAs and they're gone. And that's thanks to the work of Jennifer Watts and John Browse from the C. elegans community that, back in, I don't know, maybe 10, 15 years ago.
And in fact, Jennifer Watts, one of her postdocs is one of the founders of the company, Trisha Brock. So,
oh, really?
That's a pretty interesting connect there.
Oh, yeah. I'm such a groupie for Jennifer Watts' work.
Well, let's jump on to the Piezoone. You've recently published in Piezoone about mechanosensitive channel and food sensation. So now we got fluid membranes sensing food, or how does that all work?
Okay, so the Piezoone story. So the family, the mammalian family is called Piezoone, and the gene in C. elegans is Piezoone. When I started my lab, I thought that it would be very easy to characterize the knockout of Piezoone, and I thought that I would have like a dramatic phenotype. And you guys made the knockout for us, you guys made the gain of function for us, and it turns out that all of the phenotypes were really, really mild. So really mild, to say the least. So what happened?
That is always a sort of an intriguing but common result, I think, that
Yes.
They don't always live up to your expectations. So how did you adapt?
Especially because the mammalian knockouts, humans and all, they have either they're embryonic lethal or they have really important phenotypes that cause diseases. So why worms have this gigantic gene and does not seem to be relevant? So one thing we found is might not be relevant for laboratory conditions, okay? Because everything, you're providing them food, you have 20 degrees, we have tried 15, 25, but never was something striking. So because we detected the expression and many others, at least 3 more labs at the same time detected expression in the pharynx, it occurred to me that if we fed a different type of bacteria, we might have another phenotype. And I remember from my days in biophysics that to patch clamp E. coli or Escherichia coli, I used to use a protocol with an antibiotic called cephalexin that makes filaments of E. coli, and then you add lysozyme and they become a gigantic spheroplasm. Well, gigantic is just, it's not gigantic, but it's big to patch. So I told my postdoc at the time, Jonathan Millet, that maybe we should try, using the cephalexin treated bacteria, because it would be like feeding a spaghetti, right? It would be like way longer than the control bacteria. And it turns out that revealed a phenotype that we were really happy. because it was the only time that we could see, oh, finally the knockout is different than the wild type, and the gain of function mutant, or what we call the gain of function mutant, is different than both of them. So that was really exciting, because it's amazing how, you have all this knowledge from previous projects, and then suddenly like they merge together. So it was really nice. And that's when we came about talking about food sensation. And now the idea would be to use this knowledge to see how dependent is Piezoone from PUFAs and fatty acids, which was the idea from the beginning, but we were never able to explore it because the phenotype was so mild.
Oh, well, excellent. Well, and now we have basically only about 6 more minutes left here. So maybe we could take a little bit of a pivot on this. Interesting that basically, eating spaghetti versus eating mush gives a very different, is literally leading to this mechanosensation phenotype. So love that. You've pivoting to perhaps a little bit different area, the social aspects in science. You have a unique aspect. You're married to another researcher, and in fact, you work together side by side. So you can tell us a little bit about some of the challenges of working side by side with your spouse and getting publications together.
So, well, yes, it is a challenge, and some people are surprised, and they're always wondering, oh, how can you resist working with your partner, right? But it turns out that we met back in college in Venezuela, and so we came to the US with the purpose of getting a PhD. Not even in my wildest dream, I thought I would have my lab in the US. Like I was always thinking of going back to my home country. And then, things got complex in my country, and we liked the style of living here and doing science. And so I have worked with mechanosensitive ion channels almost since the beginning. I joined my PhD at Vasquez Lab, and he was working with potassium channels. So we are always like working in similar things, but we're always trying to learn different techniques. So we address questions using both of our brains, right? So then for our postdoc, when I moved to Miriam at Stanford, he moved with David Julius at UCSF. So he learned a bunch of things about sensory neurons and nociception and heat sensation in mammals. And I learned a ton with Miriam's in C. elegans. So when we joined the lab, we started to, all the questions we couldn't answer before, because it was not part of our labs before. We tried to, combine all of what we knew to answer questions. So for example, we have a paper it came out in January this year, I believe, where we answered with 3v1 expressing C. elegans, thanks to the work of Cornelia Barkman. We made I would say a better one with you guys. Hers was, back in the day, like, I don't know, maybe 15 years ago. So we tried to answer all of the questions we had by combining our expertise, and we determined that phosphoinositides turned out to inhibit 3b1, at least in our experimental conditions. So it's really nice because we have similar and different questions, and we have learned so many things away from each other that we can complement our science. So the lab, we run 2 labs, but again, we share materials, we share reagents, instruments, and it's like a big group, let's say.
All right, that's awesome to hear. It is fantastic. I think that is, as we all know, is a big driver. NIH loves to hear about collaborative activity between people and who you bring to the table. And it's fantastic to have a lifelong partner that is also a strong collaborator, bringing a unique and different background to
and at the personal level, if I may, we have a son, right? So it's really, no, I wouldn't say really easy, but it's easier, I would say, in comparison to other families, because we can work from home or in the lab, and we can take our son. It's a continuum. Some people don't like it because the line between work and home is not drawn very well. But I love what we do, and I think he loves what he does too. So I see it I love it. I really like working with the things I'm doing right now. So it's fine.
Well, I think everybody can appreciate the blurred lines these days with COVID. We're all, here I am in my own house running these interviews. And so there is a lot of blur these days. So I think we all understand that work-life balance, it can be a challenge, but it also can be enabling. I'm not spending a lot of time traveling from here and there. I'm actually getting a lot more done. I'm sure most other people, especially when, if you're doing a lot of writing and so on, you're getting all that stuff that you wanted to get published, get it done. We basically have another 2 more minutes for this. It went pretty fast for us here, and we've talked about basically some at least the piezo is one specific example in the mechanosensitive channel. What's some of your future directions you want to be going into? Where do you see sort of your future implications, real world implications for your research, and where you want to go?
So one of the things I would like to do is study and tackle the mechanism by which fatty acids and the membrane modulate these piezo channels. Most of the publications we have, although are really quantitative, they are not providing any mechanism because this channel is very, very big. It has 38 transmembrane segments. So tackling the sensor of the membrane tension or things like that would take, a huge amount of effort from the community. So I would like to have a molecular mechanism. In the meantime, we're doing some experiments towards that, but we are also making some lotions to use these fatty acids to inhibit as much as possible pain. So for that, we're using mice behavior. And that's one of the things that my lab is really concentrating the efforts on. And hopefully next year I would have some, like a roundup story to publish about that, because I think this is the first time I have something that has some translational capabilities, so which was surprising because I was not looking for it, but I think it would be, it is very exciting to, move away also from the mechanism towards more translation. But needless to say, we will try to understand how the membrane modulates this piezo family of ion channels. I cannot hear you, Chris. Oops.
Sorry, so I had a typing over there. You can hear me now. Very interesting. So, the translational aspect to the research you're doing, you might be moving in and becoming an entrepreneur, coming up with the lotions. And I have a question from one of our members in the audience. Are those lotions for human use? And I presume so, right? You're thinking.
That's the dream. That's the dream. Yeah, I have partnered with a collaborator at NIH, and we're working on it. I cannot give too many details because we don't have too many details. It's not that I want to keep a secret, but I don't want to, jinx the project. But that's the goal. That's the goal. And I assume they would be safe given the composition because it's just fatty acids. And I think our last paper on that was really obvious towards that. I mean, like essentially the last paragraph of the discussion was, the goal is to use these to what it's called to ameliorate mechanical allodynia or mechanical pain, right? So.
Well, I love it. And you know what? We burned up our time here, and I love hearing how your story is leading towards a translational impact. Almost sounds like almost a natural nutraceutical is lotions being developed. Very interesting area. I love it. All right. Well, thank you.
Went very fast. Thank you, Chris. It was very smooth and fast.
Yes, you're welcome. Well, thanks everybody for attending, and thank you, Valeria, for speaking to us about your wonderful research. And I'm excited to hear more about how this all develops down the road. And thanks everyone for attending.
Thank you so much.